So now let's see the three versions of triploblastic animals. We'll start with the easy one, which we saw when we are doing an example of triploblastic. And this is just this guy, where you have we say the innermost thing, what is it? The gut. Wow, I know everybody got it correct. And then the lining, the wall of the gut, this red pigment is the endoderm, okay? And then what about the orange one? It's the mesoderm, the in between. And then what about the last one? It's the ectoderm, the outside. You must be able to draw this as well, because the question about drawing and explaining does come. Now, but there's something I want you to see. When you are dealing with this type of guy, is there any space between the three structures, between your endoderm, mesoderm, and ectoderm? There is no space. You can see that all the space has been taken by the mesoderm. Okay, so the endoderm is stuck to the mesoderm, which is stuck to the endoderm. There is no space here. That means there is no cavity. And if there is no cavity, that means this guy does not have a coelom. He's the first type of triploblastic animal. So we have the statement which is saying mesoderm fills up the whole cavity. The whole space between endoderm and ectoderm is filled up by the mesoderm. There is no space here. Okay? So it fills up the whole cavity between endoderm and ecto. So between endo and ecto, there is a meso, which is filling up the whole space. And there's no other space. So in here we have no coelom, no coelom. Huh? Don't forget, it's coelom. That's the first type of triploblastic animal. And we'll call it an acylomate. Okay, acylomate means there is no coelom, right? And if you look at it here, we're going to put our animals. Remember, we have six animals that are triploblastic. So we want to see which ones are acylomate, which ones are whatever. So the first one is this triploblastic acylomate. They are also just called acylomate animals. And that is going to be our platyhelminthes only. He's the only one that is going to be this. And you can see it right here in this diagram. He is our flatworm. Huh? Can you see it? This is the flatworm. You see how flat it is? Now, if you have a look at this inside, can you see here is the gut? Okay, I'll put the gut in red to make sure you can see it. Put the gut in red. Here is the gut. And then the yellow is going to be our endoderm, right? And then the pink is going to be our mesoderm. And then the blue is going to be our ectoderm. So is there a coelom here? Do we have any space? No. So it's an acylomate. And which phylum belongs there? When it's one, we say phylum. When there are many, we call them phyla. Okay? And what we are doing is called phylogeny, which means grouping animals by their phylas. Okay? Remember, phyla, plural. One, phylum. So we are talking about the phylas, the animal phyla. Okay, the animal, so it's not, so we are talking about the phyla. Let me not say phylas, but the phyla, the animal phyla. Okay, platyhelminthes is one phylum out of the six and is the only acylomate, triploblastic and acylomate. Don't forget that. Now we go to the second, which is this one called pseudocoelomate. It is also triploblastic. Let's see what is important here. This outermost layer is going to be our ectoderm. We know that. This outermost layer is going to be our ectoderm. And then in here we have the space, which is called gut. Then the lining of the gut is the endoderm, right? The gut wall, the lining. Then the one that comes after is going to be what? This orange one, we saw that is the mesoderm. We saw that the orange one is our mesoderm. What is the blue one now? Which is between endo and meso. What is that blue one? What is this blue pigment? This blue pigment is what we call the coelom. So here we have our coelom. But when you're naming it, be careful, because coelom, we said it is the cavity in the body that is completely surrounded by mesoderm. So that means this should be, it is surrounded by mesoderm on the outside, but on the inside as well, we want to see mesoderm so that this blue is in between. We should have this whatever it is, the blue one like this, let's call it the blue or whatever it is. This should be mesoderm and the outside should be mesoderm. Then we know it is completely inside and outside. It is covered by mesoderm. But look at this one. Yes, it is there. The coelom is there. But if you see it, sorry for that glitch. The coelom is there, but if you see it, the mesoderm is only on the outside. It's only on the outside. So that makes it not really a coelom, but a pseudocoelom. It looks like. So we call this a pseudocoelom, pseudocoelom. That is our pseudocoelom. It is a cavity, it is space, but we have the mesoderm only on the outside. We don't have it in the inside as well. So that means it says gut, then endoderm, then some space, then mesoderm, then ectoderm. That is the pseudocoelomate. Okay? Pseudocoelomate. Who is it going to be? He's going to be the nematoda. Nematoda, what are they? Nematoda, these are the roundworms. They don't have any segments. They are just round and smooth. They are roundworms. Roundworms. And they are smooth. And all those roundworms that are smooth, few fall into this nematoda. And that's the only pseudocoelomate. That's the only pseudocoelomate. If you look at this diagram here, is a nice smooth roundworm over here. And then if you look here, we have the outside, which is the ectoderm. We have this pink, which we said is the mesoderm. Then we have this space inside. It is the coelom, the coelom. Then we have this yellow, which is going to be our endoderm. Then the inside is going to be the digestive tract, is the inside here. That's the digestive tract. But this yellow outside, this is a bit of a mistake. This yellow outside is endoderm. Okay, endo is the wall. Then the inside, that is where you have the digestive tract. Wow. So we said who is there in here? Nematoda. Who is there in the other one? Platyhelminthes, flatworms, right? These are the flatworms. Here we have the nematoda, which is the roundworms. Great. So now let's see the last type, which is triploblastic coelomate. Now we are talking about animals that are called coelomate. We saw acoelomate, which is A, then coelomate, which means they have no coelom at all. Okay, we saw them, and we saw that the outer, the in between, and the inner are stuck to each other, no space between them. Then we saw that one where the space is between mesoderm and endoderm, which is the mesodermal layer and the endodermal layer. Now we come here, the last one, coelomate. What is it that makes it now be called a coelomate? If you then look, we have our nice outside ectoderm. We have no problem with this. We can see it. This orange, our mesoderm, we can see it. Then this red, our endoderm, we can see it. Then we have our digestive tract or our gut. We can see it. But what is this blue thing then? This is our coelom, the coelom. Why can we call it a coelom instead of a pseudocoelom? It's because look on the outside is mesoderm. Look on the inside of it here is mesoderm. So it is completely, let me put mesoderm in green. Look, it's on the outside, it's on the inside. So it is completely surrounded by mesoderm. That makes it a true coelom. That is our coelomate, right? This is our coelomate. And it's going to be those four remaining guys. We've put Platyhelminthes, we've put Nematoda. So who are we left with? Arthropoda. Who else? We've talked about Arthropoda. We have Chordata. Who else? Echinodermata. Okay, Echinodermata. Okay, somebody will say Echinodermata. Okay, and then what's the last one? Oh, and the last one is that nice type of worm, which we haven't put. Annelida. And that's the segmented worm. Remember that this is the segmented worm. Right? This is your segmented worm. and you have it. Now, when you talk about these guys, the say arthropoda, you have insects here. Okay, you have your insects, you have your spiders, you have your crustaceans in here, right? You can check them up. You can check them up. They are arthropoda. Chordata, we talked about them. Those big five, huh? The big five, which we said mammals, reptiles, amphibians. We talked about them, right? Fish, right? We talked about all those. Now, and birds. Now, the echinoderms, these are the starfish. We talked about starfish in the first video. You can check about it. Then annelids are the segmented worms. So, worms. So this is beautiful. Look at how beautiful it is. So mammals, etc. Right? I'll say mammals, then etc. Right? These are the things. If you look at this picture here, we have a segmented worm. Can you see this segmented worm? So for examples that we've been using, all of them were worms. The first one was flat. The second one was round. No segments. Here is a round one with segments. And if you open it, this is what you would have seen. So not really right now, but when it was going through its embryonic development. Okay? So this is what it would have. But the coelom will always be there even when it becomes an adult. This is the outside one. This is the mesoderm. This is the endoderm in yellow. Then the space here, which I'm putting in green, is your coelom. Okay? Which I'll put in blue here. The space is your coelom, and here. True coelom, right? So we actually call it coelom. Now, what you should know, the last thing which I would like you to know as well, when we talk about these guys, is that first point is that these triploblastic guys, they are when we talk about their symmetry, we saw that diploblastic are radial. They have radial symmetry. Now, when we talk about triploblastic, what type of symmetry do they exhibit? It's called bilateral symmetry. When we talk about bilateral symmetry, we are saying these are things that will actually have a head. Okay, these triploblastic guys, they actually have a head somewhere. So they have your, when we talk about this, right? And so they actually have a head somewhere. Now, heads can be different depending. For things that stand upright, it's different from these ones that face the front, right? So when we have this, always the front of anything is called the anterior. So here the head is in the anterior region. For us, our anterior is not where the head is. The head is on the top of our bodies. But for these animals, it's in the front, right? So for us, the head is, we call it at the superior end. Okay, but this one, they're at the anterior. But anyway, they have a head, which means they are cephalized. So these triploblastic have cephalization, right? Cephalization, what do we mean? They have a point where your senses are concentrated. The nerve cells, they are going to the head. That is where everything is going and being processed. So this is where everything is concentrated. So that is cephalization. They have a definite head, and that is the one that controls the movement, right? So if the head is controlling the movement, it makes these guys active. These guys are not like those things that don't have heads. They are active, right? These triploblastic animals, they are active. And this is very important. It helps this activeness, being able to move. We use it for three things. One is to look for food. Okay, look for food. And then two is to escape. So look for prey, that is a predator, looking for prey. Escape predators. Okay, we can say predators. And then the last one is just to understand or explore the environment. Explore our environment. So just to move around and see things, right? This is what our activeness helps. So we are not passive, but it is active. One is to look for prey, and the other one would be to escape predators and to explore the environment. So what do we have triploblastic animals being? They are also bilaterally symmetric. So they have bilateral symmetry. Now, that means bilateral symmetry goes hand in hand with being triploblastic. Don't ever forget that part, right? If you know that, then you've gone along. All right. Now, the last thing I wanted to make sure you know is that there is a question that could come asking you to draw. So you must as well, you must also be able to draw these things. You must be able to draw, um, you must be able to draw triploblastic acylomate, pseudocoelomate, and coelomate. Okay, so we should be able to do that. Let's try and do it right now. Here is an example. We should be able to try and draw. So let's try with the acylomate, a coelomate. Acylomate is the easiest, right? You draw the outside wheel. Let me make it nice. Outside wheel. Then, this is too thick. Let me just draw my ugly circle. I'll put the inside. Then I go straight to the most inside. Here is the gut. Then I put my endo. So now what I'm going to do is what? Right now I've put the gut, and here I've put the endo, then here I have the meso outside, and what's left is now to show my in-between, and that is going to be the coelomate. So now as you're going to put your diagrams, you can put whatever. So I'm going to show them by having them be different designs. So this is going to be my endoderm, right? Remember, it's not endoderm, it's endoderm. Then now I'm going to put my mesoderm, right? My mesoderm is going to come in, and I'm going to give it a different design. This is my mesoderm right there. So I can put my mesoderm. And then this outer thing is going to be my, whatever I want to do to it. This is now going to be my ectoderm. So if you want to shade it like this, just a little bit, I'm not sure, whatever you do. So I've shown it like this. And I'll say this is my ectoderm. This is my mesoderm. This is my endoderm. And here is my digestive tract, okay, which is the gut. Okay, the gut is also the digestive tract. And then, now that we have the acylomate, we can put our pseudocoelomate, right? Oh, lack of space. Let me remove the labeling. I'll put it later. Let's put the second one. Here's our second one. We put this, then we put the layer. Then we go right to the center. We put this one, then we put the second layer. Right. Now, what do we then do? Remember that we are then going to have another round and then some space between. So it's going to be like this. This is what it's going to be like. Then we can call this our gut. Then you can start to shade the same way, similar way. You can put things here, make it shaded or whatever you do, right? Here is the shaded region. Then this next region, I put these W's, right? Put my wheels, nice W pattern. Here is my, make them big, okay? Make them quite big. And then now this last space, I can shade it as well. Okay, put whatever design you can put, but make it be different from this in between. So I can shade it as well. For me, it's faster. I've shaded, and ooh, doesn't look so pretty. Either way, I've shaded. Okay, try it to shade. It's like this, right? And then, so I was just trying to show you how to draw. This becomes the ectoderm. The one outside is the ectoderm, right? Then what do we have here? Mesoderm. Then what do we have here? The cavity, the pseudocoelom, coelom. And then here we have the endoderm and then the gut. That's all. Then we do the last one. Let's see the last one now. The last one is going to be like this. and then we put another one outside. Okay, and then we put, look at how you do it now on this one. This one you have to be careful. You put your nice wall like this. Then now you put something that comes like this. Look at it. It comes around from this halfway line. It goes right around like you're making another one. Then it gets to half. Then it comes like this. Then you do the same thing. Okay. You go around. You get to the center close by. Almost. You leave some space, right? You go up. You go around. And it's like this. Now, if you've done it, then what you should just do now is to shade. So you go ahead and you shade this part in between. All this part that is empty, you have to shade it. All this should be shaded. Okay? All the space that was empty inside here should be shaded. We have all the space shaded like this, and this becomes our mesoderm. And then we have our coelom. Now what do we do inside to the endoderm? Endoderm, we can try to do another design, which is the M's or something, trying to make it different. You'd have to make it look different. You can do shade a little bit, leave some space, 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 shade a little bit, leave some space. Like this. You see? We've made a different design. Wow, that's nice. That's top. You could do the same design outside as well. Leave some space, shade a little bit, leave some space, shade, leave 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 space. Now this outermost is going to be your ectoderm. And then this inside is the mesoderm. Okay? Then this space is your coelom. Then this inside is the endoderm, and then the empty space inside is your gut. And you have done everything correct. We'll say bravo. So yeah, this is the endoderm. This is the mesoderm. And this is the ectoderm. We can label this other one to say in blue, this is the ectoderm. Here is the mesoderm. This is the pseudocoelom. So I'm going to put it here and say pseudocoelom. And then here is the endoderm. The most inside. And then we have our gut. So this is everything, and you're done. You've seen how to draw, and you are done. Okay. Last thing though, we've seen that these have bilateral symmetry. What are they called? Triploblastic. Now, we didn't talk about porifera. Porifera, we said it does not qualify in diploblastic. It does not qualify in triplo because it has no true tissues. So that means it will never, ever be considered in those germ layers because it can never have them. Now here, when we talk about it, you say, what about its symmetry? Because I've seen that diploblastic have radial symmetry, and I've seen that triploblastic have bilateral symmetry, which means it can be cut in half like this, right from the head going down in this line, only in this line. That is the only line of symmetry, right? That is this only line of symmetry. That's what I mean by bilateral. The left and the right are going to be mirror images of each other. Now for porifera, it has no symmetry. There is no symmetry because you can never cut and get the same thing on both sides. If I do it like this, I have three long ones, but here I have two and a short one. So they're not the same. We can never make them look the same. This is an example. These are sponges. This is an example of porifera, right? They're sponges. These are the ones we always use as examples. They have no symmetry. So porifera have no symmetry, and they are not even diploblastic or triploblastic. They don't have any germ layers. Now, diploblastic have radial symmetry there around. We saw that the jellyfish and the stuff, but we said don't include starfish because it's an echinoderm. Then now we saw the last one, which is bilateral, which is every other animal, human beings included. The mouse, the cattle, all these, they only have one line of symmetry, which is exactly halfway like this. Anything, you name it. Now here's the last diagram which shows you. So here we have our fly again. This is an insect, some form or whatever it is. Beetle or whatever. This is bilateral. Bilateral symmetry, right? Then what about this one? This is a coral because we can see the body below and then all these arms like on top. And these arms are used to catch food as the food is moving in water, right? Because either they can move with the water, slide with the water, or they can catch the food as it's moving. So anyway, these arms are up here, not down here like jellyfish. Jellyfish has the body here, then arms down there like that. So this is a coral. It's in reverse. And still it has radial. It's a cnidarian. Remember that. So this is radial symmetry. It's diploblastic, guys, right? Radial symmetry is on diploblastic, the cnidarians, right? Diploblastic, we know that only cnidarians are diploblastic, okay? The jellyfish and the corals. Then here we have our porifera, which has no symmetry, absolutely no symmetry, okay? Absolutely no symmetry. It's not diploblastic, it's not triplo. Bilateral is for triploblastic. And this should stick with you. Now, if you know that, great, bravo. That's going to be the end. Okay? That's it. Goodbye for now. See you in the next video.